Crystalline water keeps coming and going in a stream of...
The concept of a network is a metaphor for not only the questions and problems that we want to solve, but the methods by which we can address their solutions. That not only do we need to understand the network underlying a disease, but we have to create a network of individuals with complementary skills to help address that question. So what we're doing is in my Department of Medicine, as of July, we're establishing a new division in the department called the Division of Network Medicine. There's no other division like this in any department of medicine of which I'm aware, and certainly in the US. And the concept behind this division is to bring teams of people together who could begin to construct the disease networks and modules that regulate these specific phenotypes that we know very well how to characterize as clinicians and as biological investigators. So the people in this division will include not only basic scientists, investigators, physician scientists, investigators who work in specific disease areas like atherosclerosis or pneumonia or cancer, but in addition, they'll be coupled to systems biologists, bioinformatics, individuals, applied mathematicians, physicists. I think bringing that pool of people together is absolutely crucial. And I have to say that we've also been successful at getting the NIH to begin to understand this kind of concept. And in fact, they now, and what makes it difficult at the NIH to understand this concept is the NIH has also been designed by institute on an organ system basis. So there's the Institute of Heart, Lung, and Blood Diseases, there's the Cancer Institute, there's the Diabetes Institute, and so forth. And those institutes rarely provide opportunities for cross-institute collaboration. So if one is thinking, as we are, about mechanisms that are common to multiple different diseases, one would expect that the funding agencies supporting those studies should also span the institutes in which those diseases are studied. The NIH is beginning to think this way. They, in fact, we have a very nice program that's been funded by them that involves our exploration of several different diseases and mechanisms that are common to them. So I think that the landscape is right now for thinking about disease in this way and thinking about the translation of that information into personalized therapies. One way I pose the strategy is to think about a pathophenotype landscape that underlies each disease. And this landscape, here in the Media Lab, I can say these things with some hesitancy. This landscape can depict pathways that define specific mechanistic outputs that are linked one to another, the constellation of which drive the complex phenotype. So, for example, let's say you're looking at a disease that's associated with the precipitation of proteins in a neuron that ultimately leads to neuronal dysfunction and death. And there are many such chronic neurodegenerative diseases that fall into this category. Well, the organelle in a cell which should get rid of those precipitated proteins that are dysfunctional is itself dysfunctional in some of these diseases, and that's the proteasome. So one can construe a landscape that involves the well-defined functions of the proteasome, which are tractable, they're limited in number. One knows all the reactions that link one of the functions to the next, and construct this landscape not just as a static architectural depiction of the reactions that govern the elimination of precipitated proteins, but also as a dynamic depiction at what happens if you perturb that landscape with a mutation in a particular protein in pathway X. How does that mutation affect the entire landscape, not just the function of that pathway within the proteasome? So it's that kind of analysis which I think needs to go forward. Another way to look at it is from the perspective of drug development. That we're, as a classically trained biochemist, we always try to focus on the bottleneck or the rate-limiting step in a series of reactions, of enzyme reactions. But in fact, the very few enzyme reactions are linear. Most of them are branched and linked to other reactions in a complex network of catalytic actions. So that if one wanted to affect the output of such a system, there are real challenges to focusing just on the rate-limiting step. And one of the major challenges, an obvious one, is that if you inhibit the rate-limiting step, you'll cause a buildup of all of the precursors, some of which themselves can have adverse effects. So rather than have that single drug target approach, one of the ideas we're working on now is looking at this landscape of reactions in a complex network and trying to design principles that allow one to polypharmaceutically affect more than one node in the network, to optimize the effect on the phenotype you're interested in while minimizing the adverse effects to other elements in the network. That's a tractable problem, and it would change completely how one thinks about drug development. I'll begin by saying that if you look at the successful approaches to developing drugs, there are 4 strategies, and I'll just talk about briefly about 2 of them. One is this target-based approach, and the other is a phenotype-based approach. So in the phenotype-based approach, as I mentioned earlier, we look not so much, we don't care about what the target is. We simply add a compound that we think will affect a physiologic response, or maybe the tumor will shrink in the patient with cancer, or the blood pressure is controlled, without caring about what the target is, or targets that this drug will have an effect on. If you look at the success rate, even in the last 20 years, at the phenotype approach versus the drug target approach for new composition of matter compounds, the success rate for the phenotype approach is twice that of the targeted drug approach. So the compounds that are used in Asian medicine and nutraceuticals and other approaches that are that do not follow the usual allopathic approach to treatments that we're used to in Western medicine, they fall under the rubric of phenotype screening, if you will, because with some exceptions through funding mechanisms that the NIH has offered over the years, there's very little that's been done with focusing on targets that these compounds might affect. And the reason is that most of these compounds are complex mixtures. They're not quality controlled. They're multiple targets, and right.
A shotgun rather than a silver bullet.
Absolutely, it's a shotgun. So it's another level of complexity. Not only are we not certain about what the targets are in the network of biological modifiers that cause the phenotype to change, but we don't know which of this complex mixture is actually doing the job, or which of these complex elements are doing the job. So I think we have a lot to learn from Asian medicine and from nutraceuticals. I think the challenge for us, because we never want to get rid of reductionism. We have to certainly apply some level of reductionism once we get to, at the very least, understanding a disease module, is understanding at least what the principal effectors are in these complex mixtures, so that we can, in a quantifiable way, understand what's causing the benefit. Recently, a group at Cleveland Clinic has published a really interesting study, which I think also addresses your question nicely. And it's a study which looks at 3 factors that influence disease phenotype, in this case atherosclerosis: diet, the bacterial flora in your gut, and your own genome. And what they found, this is Stan Hazen's group, what they did was they did a metabolomic analysis of patients with known vascular disease, atherosclerotic disease. And they looked at several thousand small molecule metabolites in plasma. And they looked at patients with disease and controls without. And by principal component analysis, identified clusters of metabolites that appeared to correlate with the presence of disease dichotomously. And many of the metabolites they found had been shown previously to be linked to atherosclerosis, like cholesterol ester hydroxides, for example, and other known bad actors. But they found a cluster of metabolites that were in the choline pathway, choline, betaine, and trimethylamine, and trimethylamine oxide. I'll come back to the distinction between those two in a moment. These are all found, derived from phosphatidylcholine in membranes, and they're richly present in red meat. And what they found was that, first of all, trimethylamine oxide is a product, a synthetic product, of the action of a flavin oxidase family of enzymes in the liver. But trimethylamine, which is its precursor, is only made by bacteria. So they found this cluster of related methylated fatty acyl derivatives and ultimately trimethylamines. And they explored the source of the trimethylamine in the guts of animals. And they took an animal that was genetically predisposed to getting atherosclerosis, fed the animal choline in its diet. This is a mouse. The animal developed much more profound atherosclerotic disease. They then sterilized the guts of these animals with antibiotics, and they attenuated significantly the extent of atherosclerosis. They went on to show that the choline is metabolized to trimethylamine by gut bacteria. The trimethylamine is absorbed from the gut into the portal circulation, taken up by hepatocytes where it's metabolized to trimethylamine oxide. And trimethylamine and the trimethylamine oxide promote atherosclerosis by, at least according to their most recent data, activating the scavenger receptor pathway in monocytes, which allow foam cells to form, which are the basic cellular substrate for the development of an atherosclerotic plaque. So this, I think, is an extraordinarily interesting finding because it links diet to the metagenome of your microbiome, and then further to your own, in this case, liver's ability to metabolize bacterial products absorbed from the gut. And it speaks to yet another element of alternative medicines, which has also become a little more prevalent in recent years, and that is, can one change the bacterial flora of the gut and in that way influence diseases? This has been tried in ulcerative colitis, for example, and there are small series which suggest a benefit to changing the gut flora. It's not clear exactly how it works, but the latest number I heard was that if you look at the source of all the DNA in you as a person, only 10% of it comes from your cells. The rest actually comes from the bugs in your gut and on your skin. So the question is, who are you?
We're an ecology then.
Well, I think perhaps a good way to address that issue is to return to the disease scenario for a moment, and to make the case that disease networks not only include the molecular determinants of a phenotype or the network of bacteria that influence the phenotype ecologically, but also the social network within which the person exists and the behaviors that are learned as part of that social network. So smokers tend to congregate with smokers, and obese people tend to congregate with obese people, and athletes tend to congregate with athletes. There is an element of learned behavior that influences not just the expression of disease phenotypes, but of healthy phenotypes as well. Vegans tend to congregate with vegans. I'm not sure that's always healthy, but that is in fact the case. So I would say that I'm not here to argue that one should ever consider eliminating reductionism. I think it's absolutely one element in the armamentarium that we have to understand complex problems in every field, be it physics, biology, or sociology. But I think one needs to try to tailor the elements of a network, social or otherwise, to a reducible module that can be tractably analyzed, and then with that analysis, its perturbation or modification be brought back to the broader network to understand the implications for the entire behavior.
Crystalline water keeps coming and going in a stream